Good morning. I want to thank the organizers for inviting me to speak in this conference and in this session. I'm going to talk about the human genome revolution and how advanced technologies really opens up new questions and new ideas and new abilities in our research. So this is a DNA sequence. This is a picture of a DNA sequence. DNA is the code of life, the book of life. It has information how to build an entire person, human being, from beginning to end. And every living organism contains DNA, its own book of life. It could be animals, it could be plants, it could be bacteria. This DNA is located in all our cells. So this is a human being composed of tens of billions of cells. In each cell, we have the exact same book of life, even though the cells look and behave differently. So if we take one cell and we schematically draw it, it could be a circle within a circle. We have the outer membrane, we have the inner membrane. This is one cell. This could be taken from the tip of my finger or toe, or I can even spit into a tube, and I will have cells of myself inside the saliva from my mouth. Now, if we can take this cell and open it and pull out one DNA sequence from beginning to end, it will be about 1.8 meters long in every cell of our body. Now, only discrete units or discrete information comes about or is expressed from the DNA. And here you can see one unit, one information, one gene is transcribed and forms an RNA molecule or mRNA molecule. It's called messenger. And then it's translated into a protein, the star over here. And the proteins are the machines that perform all the activities in our body. So let's imagine the book of life, and if we want to understand it, we could read it from beginning to end, every letter in every book, and we could write it down. And then if we want to find a change in this book, we could read it and compare it to other books as well, and then we can identify whether an A turns into a G or a T into a C. If we want to understand it even more, we should read the information expressed from our DNA. So these discrete units come out of the DNA and become RNA. So we should look at the messages, the recipes as well, and see whether they are discarded or whether they are intact. So now if I just use biology or genetic language, the changes on the left-hand side would be genetic changes. So a typo in the book of life would be a genetic change or a genetic mutation, and a change in the signal or the information which is expressed on the right-hand side would be called epigenetics, or outside of the genetics. So the human DNA was read from beginning to end in 2001 for the first time. From beginning to end, there's about 3 times 10 to the 9, 3 billion building blocks in one DNA sequence from beginning to end in every cell of our body. And in order to understand it or to reach this goal, we use the first generation sequencer. These are the ones on top here. They look like a small refrigerator. Large universities like MIT and Washington University and Cambridge in the UK bought hundreds of these machines. It's called the first generation of DNA sequencer or DNA readers. They were developed at the end of the 1980s and used even up till today. However, the big leap in our understanding or abilities is the box below it. This is the second generation of sequencers. It took a very long time to read one DNA sequence of the human genome on the first DNA sequencer, and now it's much faster. So if I make an analogy of this leap between the first generation sequencer and the second generation sequencer, I would say the first one is like a car from about 100 years ago that would drive at the speed of about 4 kilometers an hour. And today the machine we're using, we're really orbiting in space and collecting billions of information, of nucleotides, of DNA sequences compared to the first generation sequencer. And this is a huge leap in our abilities to understand biomedical advances. Now the thing is that the evolution of the machine didn't stop there. What we have now in our lab is this machine down here, which is the size of a mobile phone. So it's completely incredible, and I'm running out of metaphors, but I would say that instead of driving the car or flying in a spaceship, now we're sitting on a meteor itself. So every time we have a new machine, the leap is about 100,000 times more in speed and amount of information we can collect. There's no other technology in the history of mankind that advanced so fast and so quickly like DNA readers or DNA sequencers. So it expands our abilities, but let's get back to basics again. I said the first DNA sequence from beginning to end was read in 2001. So at that year, a person could choose whether to buy 4 jumbo jet airplanes or to sequence their own DNA. That was roughly the same cost. And today, we have to decide whether to buy a bicycle or to read the DNA, because it's very, very accessible. It used to cost $1 billion about 15 years ago. Today it costs about $1,000. It's accessible, and the question is, of course, what are we going to do with all this information? You know, you could even sequence the food that you're eating. We said that DNA is everywhere around us, and this is a layer of information we haven't accessed before. DNA is everywhere. I am now scattering my DNA here around me. The DNA is on your bottle, the DNA is on your computer. If we have these machines that will give us the ability to read DNA everywhere, we'll start collecting it. We keep on asking ourselves what for, but I'm sure we'll find uses for it. This is Nick Volker from Wisconsin. This is a child who suffered from hundreds of operations up to the age of 4. This image was taken 7 years ago. The doctors didn't know what was the cause of his disease. And 7 years ago, when DNA sequencing cost about $100,000, they decided to take this unprecedented and unproven technology and read the DNA from beginning to end. And to his luck, they were able to locate the exact mutation that caused the suffering of Nick. And he was lucky enough that this mutation came about or was expressed mainly in the blood cells. So just by bone marrow transplantation, they changed his DNA, and this is how Nick looked like a few months later.
Wow.
So this is the example, the first example of how DNA sequencing can save someone's life. But we see DNA sequencing also now in recreational forms. So some athletes want to read their DNA to see whether they can reach their extreme abilities, or maybe they should avoid from extreme sports if they have some mutations that could lead, for example, for cardiovascular complications. And if we want to understand a person's health and disease, we have to look at the genetics, and we said that we have to look at the epigenetics, the expression of these DNAs in particular cells. And 2 other factors are the environment, because we carry a lot of microorganisms in our body that can affect our health as well. And the final point we have to look at is our lifestyle that can also affect, obviously, various diseases. So this is the team in my lab here at the Faculty of Medicine that performs many of these experiments, and I'm going to tell you about a few experiments that we carried out using these advanced DNA and RNA sequencing technologies. So the first one is on a mental disorder called schizophrenia. And what we did is, instead of looking at the DNA, at the genetic effects, we wanted to see the epigenetic effects. And we took 20 schizophrenia patients and 20 control, together with our collaborators, Professor Ruth Navon and Professor Akira Sawa. We reached the upper nasal cavity. We took a few biopsies from the top of the nose. This is the closest you could get in a person when you want to retrieve some neurons from them. So it's about 3 centimeters from the brain. 20 controls and 20 schizophrenia patients. We profiled millions of RNA molecules. We wanted to see what is the difference between these groups. And here on your right-hand side, you can see that we found 1 RNA molecule which is highly elevated in the diseased versus the control group. So by using this technology, we were able to find the first molecular marker of a brain disorder. This was a very small set of patients, and now we're trying to expand it to a much larger set. Another experiment was carried out on this lady on the left-hand side. Do you know who she is?
Henrietta Lacks.
Of course, yes. This is Henrietta Lacks. Henrietta Lacks died from cervical carcinoma more than 60 years ago. I think it was in Baltimore in the United States. And her physician took the cell from her cancer and kept on growing it in a plate. Now these cells were propagated and they're spreading every research lab around the world. So we took these cells and very naively we wanted to ask, what can we find in these cells 60 years after Henrietta Lacks has died in terms of DNA and in terms of RNA? So we wrote an algorithm that could really find the small snippets in these cells, and I'll show you the output here at the bottom. So it says here that the most prevalent non-human RNA that we found in her cells was human papillomavirus type 18, HPV-18. So this might be reminiscence of the cause of Hila's cancer and Hila's death more than 60 years after she died. So this is an immense, powerful technology that we could use to read the history of a person's health, or we could also read healthy people and try to understand predictive or preventative medicine. And the final study we carried out is on Alzheimer's disease, deterioration of the brain. We performed this together with Professor Uri Asheri here at the university. It was known for many years that enriched environment can postpone the appearance of the disease. Now, the exact mechanism was unknown, so what they did is they took mice and they grew them in enriched environment, physical and mentally, and those that were in regular cages. And a few weeks later, we profiled the different sets of RNAs in the brain to understand what caused or what was elevated in these enriched environments. And we were able to find the exact molecules that have changed in the brain based on just lifestyle of these mice. They were in enriched environment cages versus normal cages. And what they're trying to do now in the lab is change or postpone the appearance of Alzheimer's without an enriched environment, just by using these very specific molecules reinserted into the brain of these mice. So, in order to summarize this, in order to understand a person's health and disease, we have to look at multiple parameters, whether it be genetic, epigenetics, environment, and lifestyle. And by these new advanced technologies is becoming extremely available in many of our studies. Another small project the student took in the lab is show how accessible the DNA is. So we said that every person can read their DNA now, but they can't really query their DNA. So an internal project we carried in the lab is called Gene Genome, and in short we call it GeneG. And it runs on these small machines that are in everybody's pocket. So if you sequence the DNA and you upload it to this app we perform, you can actually start querying your own DNA, because we carry our DNA everywhere and all the time, right? But you can't ask your DNA questions. So now if you use our app, which is not available due to legal issues, only in the lab, you could upload your DNA and you can start querying your DNA questions, and you say, hey, my DNA in this particular location has a T. How does this compare to many other genomes that have a C there? And by comparing DNAs, many DNAs and many clinical features, you could understand your DNA better. And potentially, a guy and a girl can bump their phones and virtually compare the DNA to understand whether they have mutations in the same genes that would be carried over to the next generation. So this is totally experimental, but it's accessible and it works. And since the technologies really bring the DNA to our footsteps, there are going to be technologies that will allow every one of you also to read and to ask questions to your DNA. So how is biology becoming more technology, and how is technology becoming more biology? So this is an IBM computer called Watson. In 2011, it won the Jeopardy contest in the US. It wasn't connected online, so it was offline, but it learned and it had intuitive answers and it won this competition, though IBM was very excited, but that wasn't their main goal, so they took it, and in 2013, they used this system. They started using the system in a hospital in New York to help physicians decide or give medications to patients based on information that it learned or keeps on learning. So it's there in the hospital, and they're trying to prove that actually the decisions of what's on their computer is at least as good as the best physicians they have there in a hospital in New York. So computers are becoming more human, but I think what is even more exciting that the biology is becoming very digital. So a scientist called Craig Venter led a team in the US to design a digital organism. So they read enough DNA in order to understand what a minimal human being, what a minimal organism requires. And then they typed this code on the computer, they pressed enter, and these 4-letter codes went into the synthesizer next door. After synthesizing the entire molecule, they took this DNA, they inserted it into an empty bacteria that didn't have DNA in it, and this organism started living and started replicating. So this was also carried out a few years ago, and this is the trend we see in biology, that biology is really becoming a catalog and digital, and you can order DNA sequences at longer and longer stretches, and eventually what they did there is inserted it into an empty cell. And many companies now are trying to synthesize the longest sequence available, which eventually, by clicking on a computer, you will be able to order, just as simple as Craig Venter did it in his experiments. Thank you.